Hello and welcome to Inside Exercise. I'm Emeritus Professor Glenn McConell from Melbourne in Australia. The brief of Inside Exercise is to bring to you the absolute who's who of exercise metabolism, exercise physiology, and exercise and health, and to do so in a way that brings rigor, but also is accessible to the masses. Today we have Andrew Coggan from Indianapolis, where he's at Indiana University in Indianapolis. Andy has an interesting background. I've known him for a long time. Indeed, he actually was one of the researchers that stimulated my interest in carbohydrate metabolism during exercise. But he also, a cyclist would know him for his work with functional threshold power, his book on training with power. Although, as you'll see, he considers this to be more of a hobby. He's a more serious researcher, as I said, carbohydrate metabolism. Also looking into exercise and heart failure, exercise and type 1 diabetes, and more recently, nitrate metabolism and exercise, which is the focus of the talk. So enjoy it.
Thanks for having me, Glenn.
Yeah, great. So tell me, how did you even get into this field of exercise?
Well, when I was a junior in high school, I had been racing bicycles for a couple of years. One of my club mates took an exercise physiology class at Ball State University. And when he was done with the class, he gave me the textbook, Herbert DeVries, Exercise Physiology is the simple title. But I was smitten by the topic. Science, exercise, math, I just loved it.
Right. So where'd you start?
Well, being from Indiana, you heard about this lab down the road at Ball State University.
Exactly.
So I actually did my undergraduate there and my master's degree. I went to University of Texas at Austin, where I got my PhD under Ed Coyle. I was actually Ed's first PhD student. Then I did my postdoc at Washington University School of Medicine with John Hollisey. And then I've been an academic vagabond ever since.
Wow, that's a stellar back. For people that know this area, this is a stellar background. Now, you also, I know you're a modest man, but you mentioned the cycling. I happen to know that you were like state champion time trialist in pretty much every state you went to there, right?
I always said I was good enough to dream. I was the cycling equivalent of a 220 marathoner. I mean, literally, right? Most people would look at that and you go, wow, you're a really good runner. But the difference between somebody who can run a 220 and at the time, the world record, 210, it's, a chasm, right? So I was good enough to dream. Far better scientist than I ever was an athlete.
Well, hey, that's saying something both ways, I think. So, I mean, okay. So he's being too modest. Indiana state champion time trials when you're in Indiana. Then when you went to Missouri, you're a Missouri time trial champion.
Yeah, I was good at time trials. So as I moved around, I won, I could win TTs. I could win at the regional level.
Okay.
But, if you went to the national level, then I was also right.
Yeah. And you know what, we're not going to talk about this. Like we did talk about what we're going to discuss today, but Andy is very well known as well to cyclists. So can you just say a very few words on that, and then we'll talk mainly about your more academic career?
Yeah, when cycling power meters first came out, I was still competing as a master cyclist, and I'm a numbers guy. I got intrigued by the data, and so really, as a hobby, started running my mouth online with various ideas about how you could leverage that data to try and improve performance, and that led me to connect with Hunter Allen, a cycling coach. We ended up writing a lay book together. I was on the coaching education program for USA Cycling for many years, giving lots of lectures, etc. Contributed a bunch of my ideas to the WKO software for data analytics, that kind of thing. But really, those are hobbyist endeavors. I never considered that part of my career. I always tell people I'm an exercise physiologist. I'm not an applied sports scientist.
Well, I think you might be a little bit of both. So I know a lot of guys that know your name from training with power meters and functional threshold power and all that sort of stuff. So I think you were being a little bit modest there. And you've also built a wind tunnel at one stage, you told me.
As a matter of fact, I did. Yeah, I built a wind tunnel in my basement. I still have it.
Cool. Okay. So how did you, so you started off doing stuff, research in regards to, what did you do at Ball State? And then how did you move up to nitrates? What was your career?
Well, like many, I first started out trying to understand my own physiology, my own performance limitations. But, unlike, and I literally said this to John Holosey when I started my postdoc, I said, unlike my PhD mentor, I'm more mechanistically oriented. I'm curious about, the human responses and adaptations to exercise, whether they have performance implications or not. I just want to satisfy that intellectual curiosity. So working in Ed's lab, we did a lot of carbohydrate feeding work and working with trained cyclists during my postdoc, working in John's lab, did some longitudinal training studies. That's where I first started using stable isotope tracers. But my postdoctoral fellowship was actually funded by a training grant that John had from the National Institute on Aging. So aging and exercise was part of the foci there.
Which tends to happen as people get older.
Yeah, well, you start out
getting older here.
Yeah, you start out studying it because it's fundable, and you end up studying it because it applies directly to you.
Exactly. We're not selfish. No. Yeah, but yeah.
So I started out in the sort of traditional kinesiology, exercise science, combined teaching, research, expectations, tenure track position. But after just 2 years at Ohio State, I got pulled back into the medical school world. So I spent the vast majority of my career on so-called soft money, funding my salary entirely off of grants, either mine or working on somebody else's project. And in that case, the way the funding is distributed here in the United States, it has to be clinically relevant questions in order to get research dollars. So as a consequence, I've worked with a lot of different clinical populations, starting out with trained cyclists and then untrained individuals and then healthy elderly, but also heart failure patients, patients with type 1 diabetes, patients with type 2 diabetes, more recently assisting with studies of cancer patients, patients with chronic kidney disease, et cetera. Largely driven by, you have to pay the bills. But one of the advantages I've come to realize is that you think you understand the physiology of exercise, and you're proud of the depth of your knowledge, et cetera. But if you've only been working with one particular population, what you think you know there doesn't necessarily apply when you move to another population. So having the opportunity to work with a lot of different groups and interact with a lot of different physicians with their specialized knowledge, I think has really deepened and broadened my understanding.
I mean, it's challenging, right? I mean, we've got to kind of, because then the people are going to, when you're giving a talk, they're going to assume you know everything about heart failure and everything about diabetes. But it's cool though, right? I mean, you learn a lot. You meet a lot of different clinicians and patients.
One of the arguments I've long made is that a really well-trained exercise physiologist is like the ultimate integrative physiologist.
Yeah.
If you're a renal physiologist, you can worry only about the kidneys. If you're a cardiologist, you can worry only about the heart. But in exercise, we're interested in the perturbation to homeostasis itself, and it impacts everything in the body. So to truly understand, you have to know an awful lot.
Yeah. So you would know as well. I remember in my when I first got my PhD, started working, it was like, oh, you're an exercise physiologist. You can teach cardiovascular physiology. You're an exercise physiologist. You can teach respiratory. This is just pure, not respiratory during exercise. Just, because you're breathing hard, you can teach respiration. Because your heart's pounding, you can teach cardiovascular. I was like, what? All
right.
Okay. So that's a good background to what you've done. So what about nitrate in particular? How did you start moving into that area?
Well, prior to my current position, I was at Washington University School of Medicine in St. Louis for 14 years. And I have a colleague and friend back there, a cardiologist, Dr. Linda Peterson, who came across a funding opportunity announcement from our National Heart Lung Blood Institute looking for studies of nutrition in heart failure. And it was just at the point that the dietary nitrate story was starting to emerge in the field of exercise. There were a total of 5 papers that had been published at the time. My initial interest, being an endurance athlete myself, was the fact that it's been reported to reduce the oxygen cost of exercise. And my thinking was, if there's any patient population out there that might benefit from being able to get more, bang for their oxygen buck, it would be patients with heart failure who have a compromised ability to deliver oxygen to their muscles. So that's what got us started studying dietary nitrate using beetroot juice, as many others have done. But that then led me into really diving deeply into the nitric oxide literature and coming across a number of studies in animal models looking at the role of nitric oxide in modulating muscle contractile function. So I suggested to Linda we needed to add that to our battery of tests that we were doing on these heart failure patients. I ultimately roped in my friend Jim Martin, who's an expert on sprint cycling power, a biomechanist out of University of Utah, and his doctoral student Ernie Reimer to do a study. But then started seeing that really the effects of nitrate supplementation are much more prominent during very short duration, very high intensity exercise versus on endurance exercise. So when I moved to here at Indiana University, Purdue University, Indianapolis, IUPUI, and no longer had a cardiologist at my beck and call, I returned to my roots and started doing studies of nitrate supplementation in ostensibly healthy older individuals, people who have reduced muscle power that impacts activities of daily living, risk of falls, etc. So it's a logical application.
I think,
and that's what I'm continuing to pursue.
As far as I was aware, I think you were one of the first to. Actually, I want to step back a little bit. So I want to get to that in a bit. What I want to do is give some background to people on, why are people. So you said about the reducing the oxygen cost, and that's been found in some papers and not others, and whatever. We can talk about that. But just the background in terms of nitrate. So when you take some nitrate in, whether it's in, as you said, beetroot and various other things, or beetroot juice, or however you take your nitrate. Can you just explain a little bit to the listeners what actually happens? So, the nitrate to nitrite to nitric oxide, etc.
Yes, historically nitrite and nitrate were viewed as either inert degradation products of nitric oxide, or worse, as potential carcinogens. But it was realized, 20 to 30 years ago, it really started to emerge. It was recognized that if nitric oxide can be oxidized to nitrite and then to nitrate, nitrate can also be reduced to nitrite and then to nitric oxide. That's aided via anaerobic bacteria under our tongue, at the back of our mouth. So it's sometimes referred to as the enterosalivary pathway. And it's been shown that at least with acute ingestion of a nitrate source, these bacteria play a key role. So if you use powerful mouthwash to, kill off those bacteria, you can limit flux through this pathway. If after ingesting something like beet juice, you constantly spit out your saliva rather than swallow it, you can block the effects.
Yeah, exactly. So we've actually used, we and others have used that as a way of studying what are the effects of nitrate versus nitrite. So you have the mouthwash that stops that conversion from nitrate to nitrite.
Right.
Yes.
Now what's emerged more recently, or has been emphasized more recently, is the importance of tissue stores. So that it's been known for 10 to 20 years that the tissues, such as the heart, have higher nitrate concentration than the plasma. And so work by Picnova, Schechter, and also work conjunction with Andy Jones, proposing that muscle acts as the body's nitrate reservoir. In large part because muscle is the largest tissue, so it becomes the largest source of nitrate in the body. In that case, I don't know if we can still refer to it as the enterosalivary pathway.
Yeah.
Because it's not being derived from the gut, at least at that moment in time. There's nothing to do with saliva. So I don't know what we really want to call it. We call it the alternative pathway, the backup pathway, etc.
It's like a lot of things, isn't it? Like, you know, the classic IL-6, they're always thinking that was coming from white blood cells, which, you know, it does. But in the end, during exercise, it's largely coming from muscle. So, you know, I think muscle's doing a lot of stuff. And that even adds to the complexity of the integrative physiology, right?
Yeah. And while we're on that, though, I want to work in a couple points. Although I, like many other people, are attempting to leverage this alternative pathway to enhance nitric oxide production and hopefully obtain, you know, physiological benefits, it's important to realize that at least in healthy individuals, about 95% of the nitric oxide in the body is actually produced via the nitric oxide synthase pathway.
Exactly.
And the amount that's actually derived from dietary sources is only about 5%.
Exactly. And that's where it's been really, for me, some of the stuff. So, you know, I've done some work on nitric oxide. I had trouble getting my head around. Yeah. Now I'm going to be modest. I've had my head had trouble at the start going, hang on a minute. So you're having this beetroot juice and it's affecting your oxygen consumption and your force and whatever. But during exercise, most of the nitric oxide is coming from nitric oxide synthase, you know, which is highly expressed in the muscle, et cetera. So that's where I had trouble. And some of my questions sort of later on were going to be like, well, hang on, how does that actually work? And why doesn't a nitric oxide synthase inhibitor have the same effect, or, you know, or a nitric oxide donor or whatever? So,
right.
So just along those lines, so as Andy said, you know, nitric oxide can be, you can take a nitric oxide donor, for example, so sodium nitric peroxide. You can take something to stop the breakdown of the pathway after nitric oxide, which we haven't talked about. So Viagra, everyone knows about Viagra.
Yep.
And indeed, you know, 1998, nitric oxide won the Nobel Prize, which is what the heading was of the. Actually, the heading of the paper here in Australia said Viagra wins Nobel Prize.
Oh, yeah.
Yeah, because nitric oxide stimulates guanylate cyclase to produce cyclic GMP, and then you've got the signaling that causes relaxation of muscle. So there's the Viagra bit to increase blood flow.
Smooth muscle.
Smooth muscle. And that increases blood flow. And so what you can do is give a nitric oxide donor. You can give something to stop the breakdown of the byproduct. It's called cyclic GMP, as you know. So anyway, all those things, I thought, hang on, but you don't necessarily get the effects that you're getting from the nitrate when you give a nitric oxide donor, or you prevent the breakdown of the byproduct. So that's where it started to sort of do my head in a little bit. So we're getting a bit technical here, but do you tend to think with your work with the muscle force and things like that, the effects you get from nitrate, so from the beetroot juice, which obviously contains other things, but we tend to think it's the nitrate, obviously. Do you get the same if you give a nitric oxide donor, or if you give something to block the breakdown of cyclic GMP, which is the byproduct downstream? Or do you know that?
I recently co-authored a review article with Leo Ferraria and his doctoral student, Ravi Kumar, from the University of Florida. And Leo and Ravi work more in animal models, particularly with the rat diaphragm. So my contribution was on the human end of things. Mm-hmm. And in reviewing the literature, there's actually a paucity of information with respect to muscle contractile function. So the real answer is we don't know, but there certainly is potential there. I mean, we do know there are other things in beetroot juice, but when we do these studies, we, like most other people, are buying the product from the company, and they provide the nitrate depleted placebo. So if you observe an effect, you can be confident that it's due to the nitrate. What you can't say for certain, though, is does the vehicle matter? Is the fact that you're delivering the nitrate in beet juice important.
Yeah.
And there's only been a handful of studies with exercise outcomes that have actually compared beet juice against, say, potassium nitrate. There's more studies looking at the effects on blood pressure. And if blood pressure is your outcome variable, it seems to be all about the nitrate.
It does. And it also implies pretty strongly that it's the nitric oxide at the end. So what I was getting at, that's having the effect. What I was getting at is, if you're doing your muscle force measurements and you're getting a certain result with nitrate, would you get the same result if you gave sodium nitroprusside, for example, so that you would know it was the nitric oxide? Because, you know, we've done studies where we infused sodium nitroprusside into the femoral artery, and then you could look at the factor.
If you weren't retired and we didn't have a pandemic, I should be planning a sabbatical where I could come down to Australia and we could test these hypotheses together. The problem is that, you know, those kinds of experiments, every sort of investigation has risk and risk benefit considerations, et cetera. So it's not the kind of experiment that's easy to do here in the United States, especially if you're a PhD like you and me versus being an MD. There are lots of mechanistic questions that you would want to pursue in human beings, and you find yourself constrained by such issues, which is why the idea, it's like, oh, I can study the effects of nitric oxide using a simple dietary treatment, you know, drink a bunch of beet juice, is an interesting, useful tool. So and to me, that's what it's all about. I want to understand how the system works by manipulating it. Other people want to know, if I drink this beet juice, will I win an Olympic medal?
Exactly.
While performance is the ultimate integrative outcome, I'm really after the, you know, what is the mechanism?
Yeah, we've actually both been through the same. So as you know, I went to Ball State as well. So with David Costa, both our supervisors. And then that was fantastic. But then as we've moved along during our career, we became more and more mechanistic. And, you know, you start off, I personally start off, it was all about performance. You know, my master's study was tapering or reduced training in runners. And then it became, with my PhD with Mark Hargraves here at Melbourne Uni, become more and more interested in the mechanisms. And you know, even though, as you say, sometimes it's for funding, you find it's all really interesting anyway, right? It's fantastic. So, yeah. So what about with animal studies? You know, do you know if they've done like the nitric oxide donors and whatever? I mean, I'm harping on about this a bit, but I'm just trying to think.
Yeah, the difficulty there, and again, this is something that Leo and Ravi and I discussed and worked its way into the paper, is that if you give a drug and you say, you know, block the effects of nitric oxide, you have demonstrated that nitric oxide, you know, can do something. Or if you give a nitric oxide donor and you increase nitric oxide availability, you can demonstrate that nitric oxide can do something. But the problem is, how do you calibrate that to physiological levels of nitric oxide? So there's a difference between saying that nitric oxide can do something and nitric oxide does do something. And so that's the problem I have with so much of the animal literature. Of course, you know, you want to see an effect, you go for the big effect first, something I learned from John Hollisey. So you're going to put in non-physiological levels of whatever inhibitor or activator, et cetera. If you don't see an effect, well, you're done. You can move on. If you see an effect, okay, now you somehow have to figure out, you know, quantitatively what it means. And unfortunately, so much of the literature, people never make the next step. There are also, you know, we could go off the deep end and discuss about all the differences between rodent models and humans in terms of nitric oxide metabolism. But at the end of the day, rodents are not a very good model of humans.
Yeah, well, yeah, like you say, it depends what you're looking at. So yeah, we looked at that. So the whole thing with nitric oxide synthase, NOS in muscle, was, you know, that it was higher in fast fibers and all that. But when we looked in humans, it was equal or higher in slow fibers. So, but the good thing,
and you're not the only ones.
Exactly. Yeah. With the NOS inhibitors, it's kind of nice. So we did a study where we infused L-arginine, and it had an effect. And then you say, well, but is it working through nitric oxide? Because L-arginine can also cause insulin secretion if you give it in vivo. So you give the L-arginine and then you give a NOS inhibitor. So you go, oh, okay, we blocked that effect. So it was the L-arginine working through nitric oxide. So there's lots of things you can do. So
well, that's lots of things you can do.
Oh yeah,
that we can't do here.
Yeah.
We do have a project ongoing right now where I refer to it as we're attempting to do our loss of function study. We've demonstrated that if you ingest large amounts of nitrate, you can increase nitric oxide bioavailability and enhance muscle speed and power.
Mm-hmm.
The question is, can you reduce nitric oxide bioavailability? Can you reduce muscle speed and power? We've done the gain of function studies. Now we wanted to try and do the loss of function studies.
Okay.
So we were using mouthwash in an attempt to deplete people's nitric oxide levels or reduce them and to see what would happen to muscle function. You run into another consideration here though, and that is the strongest mouthwash available for hexadine is over the counter in Europe, but it's prescription only here in the US.
Okay.
So we're using the strongest over the counter mouthwash. And as it turns out, as you might expect, if our dietary sources are normally only 5% of our nitric oxide, then, and mouthwashes are not 100% effective, well, we had no impact on nitric oxide bioavailability and no impact on muscle contractile function.
Yeah.
The good news is that, you know, athletes don't have to worry about, oh, I used mouthwash, it's going to impair my performance.
There you go.
So there is something has been learned, even if from a negative result.
The other thing is you might get the effect at rest, so you might get the effect on blood pressure going up or something. But then, as you say, when they start exercise and you're getting the nitric oxide produced from the NOS as well, then you really could be swamping it. Is that what you think might be happening? Yeah. Yeah.
Or you could think of other combinations. For example, what if you put people on a very low nitrate diet and use mouthwash? You know, could you significantly deplete nitric oxide stores?
You know that guy Raul Bescos? He was a Spanish guy.
Yes.
Yeah. So he did the VO2 max, early one of those studies, and he was in my lab for a bit there.
Yep. I've traded a few emails with Raul.
Oh, there you go. Oh, yeah, of course. Yeah, I knew that. Anyway, he's been doing studies with vegetarians. I don't know what he found in the end, but the idea there was he was thinking, hang on, what was the logic? That was hang on, they shouldn't have low nitrate. There's some idea that they have lower nitrate, right?
I don't know about lower, but you could certainly envision how, you know, oral microbiota might be different.
I'm getting mixed up. Yeah, I've actually messed that up. I think that was more the oral microbiota.
Yeah.
Yeah. Okay. So just I mean, we've talked around it, and you know, you've said you've looked at this and this and this, but what actually have you specifically found with your patients? So when they take the nitrate, or even your healthy people and your patients, what effects do you get on their force? Is it power? Is it fatigue, or what is it?
We've demonstrated in healthy, young, and to middle-aged men and women, in athletes, in patients with heart failure, and in healthy older men and women, that an acute dose of dietary nitrate, 10 times what you might get in a single day normally, will increase maximal muscle speed and hence power by about 5%.
Wow. Okay.
Tends to be a larger effect in the patient groups that may be depleted or deficient in nitric oxide. Not everyone shows a beneficial response or seems to show a beneficial response, but about 3/4 of everybody does. Okay. And interestingly, women seem to be more likely to respond than men. They don't necessarily show a bigger improvement, but they're more likely to show an improvement.
So I wonder if I should know this, and I kind of did know this, but I've forgotten. Do women have lower NOS in their muscle? So maybe they're getting less from NOS and getting more from...
I don't know about NOS, but it has been reported that plasma nitrate and nitrite levels and breath nitric oxide in some, not all studies, have been reported to be lower in women.
Okay, so that fits.
Female sex hormones may also impact nitric oxide synthase activity. As well, in older women, there is specifically a reduction in myosin light chain kinase activity, and hence myosin light chain phosphorylation, which is a potential target of nitric oxide. So at least in the context of aging, you can make the hypothesis that women should be more likely to respond because of a specific deficit.
Okay. So this is interesting, right? Well, obviously it's interesting. I mean, hey, your work is always interesting.
And it's fundable. Oh.
It is fundable. Okay, interesting scientifically, because, you know, you think about the original idea was that there was nitrate was making you more efficient, you know, less oxygen use at the same work, which is actually pretty remarkable when you think about, you know, ergogenics. But so that's more like a slow twitch thing, right? Oh, sorry, a more aerobic endurance type thing.
Right.
When you're finding that you're actually finding the force, which is obviously, I mean, you're going to be using
power,
all fibers.
Power.
Okay.
Power.
That's explosive stuff, right? So we're not talking about the slow sort of aerobic pathways, the anaerobic mainly. I have to always say that because my students will say that's anaerobic, that's aerobic. No, mainly anaerobic. So what do you make of that? And do you think it's maybe fast fibers it's more affecting? Or?
That's been hypothesized, but my starting point would say, you know, although, you know, I've operated in the world of cardiologists forever, right? And you'd tell them you were doing studies of, you know, dietary nitrate, nitric oxide, muscle contractile function, and their response is always, well, what about blood flow? You know, we're measuring muscle speed and power during, you know, isokinetic contractions that may be over in, you know, 100 milliseconds. The muscle doesn't care how much flow is happening during that time period. But because they're cardiologists and their background, this is their perspective, their bias, etc.
As a delay, that's the thing we haven't probably said enough, that nitric oxide, when it came along, well, obviously it's been around forever, but when it got recognized in 1998,
identified,
it was for its cardiovascular effects as a vasodilator.
Right.
I guess we just have to get that out of the way. So that's what.
But in fact, it's involved in numerous pathways. Absolutely. I describe it's a promiscuous molecule.
Immune function.
Right. So you know, when you we carpet bomb the body with huge amounts of nitrate. Yeah. And then the body decides how much nitric oxide is produced in what cell when. We have no control over that. There's really no reason a priori to expect that every physiological outcome would necessarily respond in lockstep with each other. In fact, if you go back, all of the early attention is in terms of performance, it's largely driven by a study out of Jones's lab where they report that, you know, dietary nitrate supplementation delays fatigue by 16%. Mm-hmm. Well, if you look at the relationship.
Just clarify what kind of exercise that was.
Endurance exercise, intense. In terms of endurance exercise, where dietary nitrate supplementation would be most likely to show a benefit, it has to be sufficiently intense. You're not going to benefit during a marathon or a 4-hour time trial. It also doesn't seem to benefit.
It's interesting because if it's reducing your oxygen cost, I mean, that's the thing, like they find with Eliud Kipchoge, all these guys, part of the reason they run so well, it's not necessarily the VO2 max, it's their percent they can hold and it's their running economy.
Well, that's the other aspect to it, is that dietary nitrate supplementation doesn't seem to be beneficial to endurance performance in well-trained endurance athletes.
When it should be.
Despite all the attention focused on it. My thinking there, it is known that nitric oxide synthase activity is upregulated by endurance exercise training. It's known that interstitial and plasma nitrate and nitrite levels are higher in endurance trained individuals. The way I think of it is every time you work out, you're inducing shear stress on your vessels and you're increasing the rate of nitric oxide production. And where does it go? Well, some of it goes to nitrite and nitrate. So you may have already topped up your stores as high as they can get if you exercise regularly.
It does make sense.
So really the individuals most likely to benefit would be those who are deficient ostensibly in nitric oxide patient populations. And this is the same argument that Jason Allen has made. And Jason was the first to actually look at a clinical population with his studies of PAD patients.
That's true.
So I really think that is the target, not, you know, the Olympian, but rather aging, heart failure,
so you would know that people with heart failure have reduced NOS.
Yes.
And so therefore,
and more importantly, and again, remember my stable isotope background, right?
Mm-hmm.
It's one thing to measure the expression or activity of an enzyme. It's another thing actually to measure flux through the pathway. So with stable isotopes, you can actually measure the rate of nitric oxide production from L-arginine, which has been done in numerous groups, including heart failure patients. So we know that nitric oxide production via the NOS pathway is diminished in heart failure.
And you know what, this just shows how complicated this gets, because I used to have so much trouble, and you probably do as well, writing grants and trying to explain that you're wanting to look at nitric oxide. First of all, they assume you're talking about endothelial NOS, which is producing nitric oxide, increasing blood flow. And I'm like, no, I'm talking about muscle, which is mainly NNOS. But the other thing is heart failure patients have increased INOS. So, you know, you're saying it's so ubiquitous. INOS is the inducible nitric oxide. Then you get bucket loads of nitric oxide formed. But then you're like at the same time saying they've got reduced nitric oxide in their muscle. So it's just really, it just is hard to explain.
Well, the body's complicated.
The body is complicated.
All you can do is hope that the reviewers are smart enough to keep up with you.
And the other thing they're doing, exactly. Well, that is a big, and that is
I said hope. They don't, yeah.
Best thing I was ever told was a guy when I was at Monash University. He said, when you write a grant, assume that the person's, you know, in a busy cafe at the end of the day, they're tired, it's noisy, and they're reading the grant, and you've got to, that's how it's going to be, because they're going to do it quick, and it's probably going to be outside their field, and, you know, whatever. So you can see why it ends up being a bit of a lottery sometimes, because it's difficult. Okay, so just to think about the mechanisms of what's going on. So how do you think it's increasing the force? Is it affecting calcium release? Is it, you know, what do you actually think is going on?
Affecting either calcium release or calcium sensitivity.
Okay.
There are two known mechanisms by which that could take place. One would be nitrosylation of the ryanodine receptor. So if you nitrosylate a single...
just to clarify, so nitrosylation is adding a nitric oxide. Do you want to just...
Yeah, the chemists would get up in arms because technically it's nitrosation, but people like to say nitrosylation to draw the parallel as a signaling mechanism such as phosphorylation.
Exactly.
But
so
yes,
make it simple. So phosphorylation is when you add a phosphate group to a protein without worrying the exact bond. So nitrosylation.
Here you're adding an NO group to an amino acid residue, serine residues in particular,
and again,
and cysteine.
In talking about how complicated things get, 15 years ago or whatever, we never even used to think about phosphorylation. We didn't, you know, like we used to, if we measured an enzyme, we thought we were a genius. And then phosphorylation became the big thing to affect, either make it more active or less active. And then you've got S-nitrosylation, you've got glutathione, which is
argination.
Argination. There you go.
Yeah.
So wow. So that's really okay, because then you've got arginine, which is part of the nitric oxide pathway as well.
So one possibility would be nitrosylation of a specific cysteine residue in the ryanodine receptor, the calcium release channel of the sarcoplasmic reticulum, which tends to lock it in the open configuration. And that would enhance calcium release or escape from the SR, and therefore should increase the number of active cross bridges when calcium levels are subsaturating. And in fact, that's what's been found, is that if you do like electrical stimulation of the muscle at different frequencies, you see increased force production at low frequencies, but not at high frequencies. At high frequencies, you're releasing enough calcium that you've basically apparently saturated the contractile apparatus. Another possibility would be nitric oxide by the classic soluble guanylyl cyclase cyclic GMP pathway, activation of protein kinase G, phosphorylation of myosin light chain kinase, which then phosphorylates the myosin light chain. And that can increase calcium sensitivity of myosin. And then finally, credit to Leo and Ravi for really digging into this end of things. Another possibility is, while we emphasize, or we classically have focused on thin filament regulation of muscle contractile cycling, there's considerable evidence now that the thick filament itself is regulated, so that the possibility exists that nitric oxide may enhance the number of myosin cross bridges that are moved away from the myosin backbone and therefore potentially available. So as opposed to being in the super relaxed state, instead they're ready and available.
Okay, cool.
To generate force. So that paper, that review paper, if people are interested, is coming out. Well, actually, it is out now in a special edition of the journal Nitric Oxide.
Okay, right.
So the difficult part, of course, is getting at these questions in humans.
Yeah.
We, especially since you're limited in the amount of tissue, and as we mentioned, there's considerable difference between rodents and humans. So really, the question needs to be answered in humans. To this end, we developed a method that allows us to measure nitrate and nitrite in 5 to 10 milligram muscle samples. It's theoretically sensitive enough to even do single fiber work when it comes to nitrate. We then worked out a method as well to measure cyclic GMP from that same extract. So we didn't have to go back to the biopsy sample and use another piece separately for that. Unfortunately, a lot of that work was shut down as a result of the pandemic, and the monetary losses that accompanied that have made it difficult to get up and going again. But it comes back to your question about fiber type. It has been hypothesized by Andy Jones that dietary nitrate specifically targets fast twitch fibers, but we've measured nitrate and nitrite in rodent muscle, the fast twitch white quadriceps, the slow twitch soleus, and we find that the nitrate and nitrite levels are 2 to 3-fold higher in the slow muscle, not in the fast muscle.
But again, we were just saying how rodents and humans aren't always the same, so that, we found the same with the nNOS. nNOS in rodents is higher in fast fibers. Right. But in we and others have found it similar or higher in slow humans.
So I currently have a doctoral student. He's in his first year. He's a direct admit from the undergraduate, so he'll be around for a while, and we're starting to kick around ideas. So I was joking with him the other day that his like I said, it was yesterday, that his part of his dissertation might consist of sitting around and teasing out individual single human muscle fibers in order to make a pile of about 100 of them, so that we have enough tissue that we could measure both nitrate, nitrite, and cyclic GMP. Tedious work, but that's what it would take.
I always like that, teasing out. Teasing kind of sounds like fun, but I think sitting and teasing out fibers is not fun. Now, okay, so you've been talking about we've been talking about the essentially usually acute or chronic supplementation. Yeah, so I want to tease that out a little bit. So I always found it a bit surprising. Sometimes you get similar result with acute, and we tried to look at this at one stage. So you do the acute nitrate supplementation, and you might and people have found you get, for example, the reduction in oxygen cost during exercise in humans, but they also find it after like 5 days or whatever. And you say, well, hang on, aren't the mechanisms likely to be different? So, for example,
well, this is a case where early experiments can sometimes mislead the field, I think. Again, the initial studies quite often entailed a loading phase, let's say 5 days. And therefore, the assumption becomes that a loading phase is necessary. And everybody replicates that until somebody actually comes along and specifically tests it. Or similarly, the animal data suggests that loading with nitrate can alter the expression of calcium handling proteins. something that hasn't been replicated in humans. So if you approach it from a clean slate, and this is, again, our interest in the heart failure world is actually developing this as an actual treatment, right? So at some point, you've got to get it out of the laboratory and into the clinics. And one of the questions becomes, well, what's the appropriate dose? How often should you take it? What's the tolerability?
Yeah.
Does it have to be beet juice? Can you use potassium nitrate, et cetera? So we did a formal pharmacokinetic study in conjunction with a pharmacologist and put together a combined nitrate-nitrite compartmental model. But it added to the literature because it's a combined model, and it's specifically in these populations that we study. But the pharmacokinetics of nitrate have been studied in healthy younger humans dating back to the 1980s or so, because of the concerns that it was a potential carcinogen. And so in a healthy younger individual, it's known that 2/3 to 3/4 of the nitrate that you ingest is cleared via the urine. The remainder, we don't really know where it goes, but most likely bacterial metabolism in the gut means it leaves in the feces as not nitrate, something else. The half-life is on the order of 5 to 7 hours. The bioavailability is close to 100%. You absorb almost all of it, but only about 5% of what you ingest as nitrate is ultimately converted to nitrite, and therefore potentially could make its way into nitric oxide. There's a whole wealth of information out there that if you're really digging into this area, you start to look at it and you go, as a drug, the big problem with it is its half-life is pretty short.
Yeah.
If you were taking a drug to affect your blood pressure, you'd want to be able to take a pill once a day, and that would carry you to the next time you take a dose, et cetera. But in this case, it's washed out fairly quickly. So in that context, you start going, well, then why should there be any need to load? It seems to be entirely an acute effect. We're not changing the bodily structure. We're going to get the effect as long as this drug is present. And when the drug is gone, the effect is going to go away. So we're wrapping up a 2-week intervention in our healthy older individuals. And they benefit with a single dose. They benefit equally after 2 weeks to that same dose. There's actually data going all the way back to the 1930s in primates, in which they were dosed in their drinking water with nitrate, and then they were periodically challenged with a much larger dose of nitrate. And for 3 whole years, there was no change. Every time you gave the large dose of nitrate, you saw the same equal dip in blood pressure. So no tolerance. That's the other thing I always hear from cardiologists, right? What about tolerance? Because tolerance develops to things like nitroglycerin. No tolerance, but also no magnification of the effect. As far as I can tell, it seems to be almost entirely an acute response.
Okay, well, there's your answer. There's my answer to that question.
Yeah. Well, you asked if you wanted to know if I knew a little bit about nitrate metabolism. So I had to study up just for this. I think it's a little more analogous to creatine. Our body makes creatine.
Yep.
Our body makes nitric oxide, which becomes nitrate and nitrite. Our body stores creatine in the muscle. Our body stores nitrate in the muscle. The only thing that's really different with if you're a meat eater, you get about half of your creatine from your diet and half from what your body makes. Mm-hmm. And if you double or triple the amount that you ingest, you can push the system to the side, right? Mm-hmm. With respect to nitric oxide, you make 95% of the nitric oxide and only 5% comes from the diet. So if you want to push the system, you have to do more than have, a big bowl of spinach. You have to have, a dose that's equal to 10 times your normal daily intake. Yep. But if you do take in these very large amounts, you can push the system.
Okay. I guess
so there are both.
Creatine is you need that loading. You need the loading of the creatine.
Well, yeah. Well,
because 5 days or whatever, and it's
you do and you don't, right? I mean, if you take 20 grams a day, after 2 or 3 days, almost all of it's spilling over in the urine. Or you can take 2 grams a day and you reach the same point after about a month.
Exactly.
So how long does it take to saturate your muscle with nitrate? A single dose seems to be pretty effective.
You know, another way, just to add to complexity, is I read a paper on you get nitrate. So you know how, I don't know if you know, but people tend to have lower blood pressures near the equator. And
oh, I didn't know that.
Yeah, there's probably various reasons, but these people found that you've got nitrate, a nitrite
solar radiation.
in your skin.
Yeah.
And the sun converts it to nitric oxide.
Yeah.
So I thought that was another.
Yeah, it makes sense.
One. So you should be exercising in the sun, and having beetroot juice and having a lot of NOS. Yeah. Okay. So
well, another study. The other question is it possible to have too much of a good thing?
Yes.
We did a formal dose response study, and in healthy older men and women, we got improvements in muscle speed and power at our standard dose, which is similar to what other people typically give. We actually adjusted it relative to body mass, so 200 micromoles per kilogram body mass, 14 millimoles in a 70 kilogram person. That's pretty much right where most people are operating. But when we doubled that, 8 of the 9 people, their muscle contractile function regressed back towards or even below the placebo value, the baseline value.
That's interesting. And that actually suggests that it is having a, again, it's a little bit, you mentioned paradox earlier. There's another thing of nitric oxide, but you talk about the L-arginine paradox, because L-arginine can be converted by nitric oxide synthase to nitric oxide. And you should have enough nitric oxide that the L-arginine doesn't have an effect, but it does. So the L-arginine has an effect. And the same there, you in theory, the nitrate shouldn't be really enough to have much of an effect, but it does. And what you're saying there is that sort of U curve is what you get with nitric oxide as well. If you have very little nitric oxide, you don't get much force, for example. The Michael Reed. If you've got just the right amount, you get beautiful force. Too much, it drops, and you're seeing the same with nitrate. So it does fit together, but it's like, hang on, I thought we had enough from there anywhere. So I guess it's like everything in moderation, right? Like a dietitian.
Yeah, the Greeks had it correct.
Mixed diet,
well, and then it does start to make you wonder. I mean, you start thinking, okay, creatine, nitrate, what other substances are out there in our diet that are having an impact on our physiology that we haven't really, recognized yet? They can't be huge effects that, if it were fatal, we'd know to not eat that plant or whatever.
Mm-hmm.
But if a human consumes a particular, food substance and it alters that person's function by 5%, well, that's not going to, influence, your ability to, evolve, reproduce, dominate the world, et cetera. Yet it's having an effect. We're all victims of our environment in a way, right? And a lot of this stuff is going on and we don't even know it.
And also, because we're so imbalanced, we could be mucking up something else that we don't know about.
Yeah.
Right.
Trying
the balance out. So anyway, just thinking about, we've talked about roughly, I try and think with these podcasts, how does it affect the average person? How does it affect health? How does it affect sport? Yeah, we've pretty much covered all those. But I guess what we haven't said is, you've said in the lab, at least, you're getting higher power. Yeah. So would you then suggest that would work for like a, 100-meter sprinter or a power lifter, or someone doing high power? Was that something that you would suggest, and are they actually doing it?
As we were talking about before, there's a fairly large amount of research now looking at endurance performance and endurance athletes, and I think it's pretty clear there that highly trained individuals do not benefit. Mm-hmm. The question then becomes, what about sprint athletes? So if you're a very well-trained sprinter, would you benefit? Mm. The early study we did with Jim Martin and Ernie Reimer, the athletes were team sport, a few triathletes. I described them as a motley crew of athletes. They weren't your classic, all marathon runners, et cetera, but they weren't highly trained sprinters. They benefited. We recently published individual participant data meta-analysis, and in that, the two of the three studies that did not show much benefit with respect to, most of these were using Wingate cycling, so peak power. We're using highly trained sprinters, one highly trained track and field athletes, and the other was using speed skaters.
Okay.
So it's possible that sprinters exercise enough, train enough, that they're producing enough nitric oxide that they don't benefit from supplementation.
Mm.
But I think it's a question that's deserving of, being revisited to try and determine, in fact, do they benefit, do they not? This does loop back to another point early on, though, and like I said, a lot of the literature and the excitement was driven by the report that time to fatigue was enhanced by 16%.
Yes.
But if you consider how shallow the slope is of the exercise intensity duration relationship, it really means they only got 1 to 2% more power.
Yeah.
Allowed them to go 16% longer.
Going a little bit longer, yeah.
Yeah. So, I mean, a 1% improvement in an elite athlete has been estimated will, double your chances of making the podium at the Olympics. So in that context, 1% is a lot, but at the same time, 1% is awfully hard to measure in a laboratory. Comparatively speaking, like I said, we typically see 5 or 6% improvement in maximal power, short duration. So I think the effects of dietary nitrate supplementation are much more prominent during short duration, high intensity exercise compared to prolonged aerobic exercise.
Right. And also, not all studies show, so, the classic idea, as we've mentioned several times, is that you use less oxygen during exercise. But a lot of studies have not shown that during the endurance exercise. So our study, we found nothing. And then looking around, we found, other studies that found no effect. So it's either small or no effect. And also, just from my personal experience, find if you don't have an effect of nitrate on oxygen consumption during exercise in humans, you may have difficulty publishing that. So I wonder if there's a bit of a lack of publication out there.
That's true. It's always more difficult to publish negative results, which leads to bias.
It goes against the predominant.
I really dug into meta-analysis, which everybody and their cousin nowadays has to publish a meta-analysis, and what I learned along the way there was how the limits of meta-analyses and how poorly so many of them are done. Meta-analysis is really designed not to tell you whether an effect is significant or not, because nobody knows what the null hypothesis should be. What meta-analysis enables you to do is better define the magnitude of an effect. Right. So, it comes back to, we're all worshiping at the great God of P is less than 0.05. Well, I could have a tiny effect, and if I measure 1 million participants, I can get, P is less than 0.05. But that doesn't mean that effect really matters in the big scheme of things. While I'm on my high horse, the other thing I have to rail against, how poorly many of these meta-analyses have been done, is that it seems like a game of one-upmanship. Well, the first meta-analysis, they pulled in 15 studies of beet juice supplementation. Well, I can do better. I pulled in 30. Oh yeah, I could top that. I didn't. I found 95. Oh yeah, we put together 200 studies. But in the process, the most important step of a meta-analysis is deciding what should be included and what shouldn't.
Exactly.
And people are pooling data from studies with markedly disparate exercise outcomes. The only thing they have in common is they gave beet juice. It's like, why do you combine those?
Yeah, exactly. And the thing you said about the small difference. So the classic thing is, like, if you look at blood pressure, so you might look at a medicine or some other intervention. You've got 100,000 people, and the blood pressure is reduced by 1 or 2 millimeters of mercury. But it's significant. But is that going to have? Is that biologically important?
Yeah.
Hey, so what I was going to do, just starting to think about wrapping it up, is there's actually a question you suggested I should include in the podcast. Is what's the study, your seminal sort of finding? You think not even in this area, just a more general thing. What's the paper you're most sort of proud of? I guess if you think back along the way, it doesn't have to be nitrate. It could be.
There's a lot of favorites.
A couple even. There you go.
Couple. Well, you know, history will be the judge of what findings were most important and what weren't.
So the citation index.
Yeah, there are various ones that I'm proud of. Actually, including, I mean, you know, sometimes it's the backstory behind them. Boy, you really stumped me here.
That's all right.
You know, as I tell my students, the studies that you do should be driven by the question, and the question should dictate the methodology.
Absolutely.
As opposed to letting your methodology and your methodological constraints dictate what questions you ask.
Sometimes you've got this great method and you want to just milk it.
Yeah. So, you know, some of the studies that are my favorites were ones where had to work really hard or had novel contributions in terms of the methodological end of things. For example, you know, looking at the effects of endurance exercise training on the rate of gluconeogenesis during exercise by infusing 13C labeled bicarbonate and measuring the rate of incorporation into glucose. Something that had been done using 14C with radioactive tracers, but had never been done using stable isotope tracers before. So working out those methods and getting that to work. Back in '93, we published.
Was that down in Galveston when you were working with Bob Wolf?
No, that was when I was at Ohio State, but it was I started using stable isotopes during my postdoc, and that's kind of what drew me to Bob's attention and why he pulled me down.
Okay, it's the other way around.
To Galveston, yeah.
Nice.
You know, one of the studies we did down in Galveston, just thinking about these things and some of the challenges, looking at glucoregulation during high intensity exercise using the eyelet clamp approach. So infusing somatostatin to suppress insulin and glucagon, and then infusing those as at replacement rates, and then infusing glucose.
I've never done that. I want to do that one day. That's cool.
It's exciting when you know, you're wondering what's going to happen. Are you going to cause the person to crater in hypoglycemia, and you're madly jacking up the rate of glucose infusion to try and prevent that? So
that I've always liked that. I've always thought that's pretty intricate stuff.
Yeah. So really, you know, if I have enough of an ego, I'd say I'm proud of many things, but I am proud of the fact that I've not let my methods dictate the questions.
Exactly.
I mean, I've used isokinetic dynamometry now because we got interested in muscle contractile function, but I had never done it except in grad school. Done stable isotope stuff, done radioactive isotope stuff, done AV balance measurements, done magnetic resonance spectroscopy, done, you know, positron emission tomography, done mathematical modeling, compartmental modeling studies. About the only thing I haven't done is any molecular biology work.
Oh, there you go.
What is everybody's bread and butter? I don't know how to do.
Exactly. I remember that when that first started, I was at Monash and everything was messenger RNA. Everyone thought it was cool to measure messenger RNA. And then it's like, so that's, you know, but then that's producing, then you end up with a protein. So it's like, oh, we're going to measure protein. And then that was hot. But it's like, hang on, you need the activity of the protein. And then you need the phosphorylate, you know, back to that. It just keeps going and going. All right. So one more sort of generic one. What makes you content? What makes you jump out of bed in the morning? And, you know, and this could be scientific, could be general.
Maybe that's why I'm still working hard, is I'm malcontent. My daughter came back from a run the other day, took a shower, sat down at the dining room table, was looking at her phone, and she said, Strava graphs make my brain happy.
There you go.
And so I can, you know, so she's a chip off the old block. She's like her mom and like her dad, right? She's a exerciser, and she's, you know, into data nerdery. So yeah, a good workout and then looking at the data, that makes me content. You know,
I didn't really say this earlier, but yeah, when you came, when was it? In 1990, I think you came down to Australia, and I first met you at Melbourne Uni. We went for a ride. We should have gone for a run. I You were just like out of there. It's like, hang on, shouldn't we ride together? No, out of there. Just fair enough. I've always wanted to run with you, so I can just go out of there.
Yeah.
Maybe
I
not.
haven't run a step in a long time.
But when you came back there and you spent some months, how long were you? Came down when I was at Monash. You came down. Anyway, we'd take the bus.
Not that long. I think both of my trips down there were only about 2 weeks at most.
Because we started doing that study with the mitochondria.
Yeah, we were trying to squeeze that study in there.
I just want to bring it together that I just couldn't believe we'd take the bus to work and back every day, and just data, just rigor, data, thinking through things, which has hopefully come out. I think it has come out. The methods, so I'm not surprised you said the methods. Not so much the result, well, the result as well, but the methodology, how does it work? Critical thinking, look at the data.
Critical thinking. Again, telling my grad students, it's like, if you know how to pipette, if you know the principle of a method, and if you are stubborn, you can troubleshoot and you can make any lab assay work, right?
Yeah.
But a lot of it comes down to, well, what is the principle? Because that tells you, well, what could be going wrong? How do I test this? And that helps you track down problems, et cetera.
Yeah, and you've got to go to the start. So even if you're talking about the pipette, are you weighing water? Are you making sure the pipette's good? So I'll have people come along and they want to show me their Western blot results of the protein level. I'm like, hang on a minute. What was the total protein? What was the this? What was this? Oh, and then we got this. No, back here. I want to know every step, Yeah. So, yeah, that's really important. And the data.
Rigor.
You also said rigor. I remember you also said that people often don't look at the data long enough. So they get the data and then they go off and write it up. It's like, no, you've got to look at the data. You've got to take the time to look at the data. Sometimes it's hard when you're under, restraints and whatever.
And this is gonna sound egotistical, like with enough gray hair between the two of us, but like, respect your elders.
I dye mine gray. No.
Scientific data doesn't have an expiration date. The people that came before us were exceedingly bright. We're the lazy ones, and they had to work really hard to generate those numbers. And while techniques may have advanced, it doesn't necessarily mean that old data are invalid. Dig back through the literature, understand why do people think what they think is true? Because so much of science exists, scientific truth exists in the minds of scientists. And you go back and you find early studies and you realize that people have misinterpreted them all along. This is something that stems from my dissertation, which is actually based on Christensen and Hansen's classic studies that were published in the 30s. Yeah, and that I'm taking up all the time here, but I had my mentors, John Ivey, Ed Coyle. They'd talk about Christian Hansen did this, and Christian Hansen saw this, and it's like, have you guys ever read these papers? Like, no, they're in German.
They're in Swedish.
So I paid a German graduate student to translate them for me as part of my dissertation, and it turns out that the experiments weren't what people thought they were. In the nitrate world, like I said. In the 1930s, people knew that nitrate dropped blood pressure. They didn't know that it was via nitric oxide, but they knew that it did that. People have been using beet juice in order to enhance nitrate availability since the 1970s. So, if you actually trace things back carefully, what you find, what you think is new is not new. But it's so helpful to know what the background really is.
Or even back to John Holossi again. I found an old paper that 1965, he used nitrate in frog muscle.
Millimolar concentrations.
Yeah, but it was massive concentrations. That was the problem. Because that was partly why, we'll wrap up now. We haven't really talked about my idea of metabolism, which is like glucose uptake and things like that. But that was why we did that study where we gave nitrate and looked at it with stable isotopes and looked at glucose uptake, because he'd done that with the nitrate. But then you go, hang on, he used huge concentrations. So again, it also, a lot of things come together. So again, with the animal studies, not only, they may or may not, sometimes they do relate to human physiology and metabolism, but often the doses that they use, as you say, they want to get an effect. So just throw a bucket at it. Okay, well, it's been great having you on here. Thank you very much.
It's been fun. We
It's
need to do it
definitely
again
fun.
sometime.
Okay, I'm
All
gonna
right.
do that. Okay, good on you. Thanks, Andy.
All right.
See ya.
Sure. Take
Bye-bye.
care, Glenn.
See ya.
Bye.